So let's first talk about acetylcholine, and in other classes, by the way, like in the psychopharmacology class, in the advanced neurophysiological psychology class, we go into much more detail on this neurotransmitter and on other neurotransmitters as well. But I want to give you sort of an overview of the synthesis, the storage, the release, and the breakdown, basically, the removal of this particular neurotransmitter, the most classical of all the classical neurotransmitters, the vagus stuff that Otto Loewy described, when he did his experiment on frog hearts to sort of demonstrate the chemical nature of transmission at a synapse. So acetylcholine, like all of these transmitters, has an abbreviation. It's A, capital C, small h. So ACH is acetylcholine, and neurons that release acetylcholine as their primary neurotransmitter are called cholinergic cells. Remember, there are probably co-localized peptides and other neurotransmitters in these neurons as well. So like all neurotransmitters, acetylcholine is synthesized from widely available raw materials, including choline, which is a byproduct of the metabolism of fats. So there's plenty, of choline circulating in the blood. There's a specific choline transporter that the cholinergic neuron will express and place in the membrane and bring the choline in. And then it's also, the other raw material is something called acetyl coenzyme A, which is a byproduct of cellular respiration, that process of burning glucose, right, in the mitochondria in the presence of oxygen, which generates all that ATP that runs these transporters. So choline and acetyl coenzyme A are actually brought together by a synthesizing enzyme, which is also expressed, in the cholinergic neuron, called choline acetyl transferase, or CHAT. So you can just remember CHAT, C, capital C, small h, capital A, capital T. CHAT is the synthesizing enzyme that grabs the choline, the byproduct of fat metabolism that's brought in by a choline transporter, and the acetyl coenzyme A that's spewed out of the mitochondria in the process of cellular respiration, and it combines them and forms ACH, or acetylcholine. So now we've got our synthesis of our neurotransmitter. We have ACH, available at the presynaptic terminal of the cholinergic neuron. And then, of course, the next step is storage, right? So we have vesicles, and in the vesicular membranes, we have in the cholinergic neuron an acetylcholine transporter. So this is a vesicular transporter because it's a protein that's placed in the membrane of the vesicles, and its job, right, is to grab the synthesized acetylcholine, utilize ATP to force that acetylcholine inside the vesicle against the concentration gradient force, to sort of store up concentration gradient force for later exocytosis of the acetylcholine into the cleft. Then upon the arrival of an action potential, there is the fusion of the pore of the vesicles, and the appearance of those pores, and exocytosis, the release of acetylcholine into the synaptic cleft. And then the breakdown for acetylcholine is actually from a degradative enzyme that is released into the synaptic cleft. It's actually called acetylcholine esterase, or capital A, capital C, small h, capital E, ache. A lot of this stuff, all these chemicals, everything else is probably giving you a headache. But anyway, ache is the enzyme that grabs onto the acetylcholine and then sort of breaks it down, right? Postsynaptically, there are, two sort of classes of cholinergic receptor that the acetylcholine will bind to and influence. And there are ionotropic, cholinergic receptors. These are, very critically important for moving muscles, because cholinergic ionotropic receptors are actually localized on the membranes of muscle cells in the periphery, right? And at the junction between motor neurons, which are efferent neurons, right, that are coming out of your spinal cord, coming out of your brainstem, and synapsing with muscle tissue, they call this the neuromuscular junction in the peripheral nervous system. It's a neuromuscular junction. acetylcholine is the neurotransmitter that's released. It binds to ionotropic, ligand-gated cholinergic receptors postsynaptically on the muscle membrane, and it depolarizes the muscle and provokes contraction. So the acetylcholine has a very, very important role in the peripheral nervous system at the neuromuscular junction. Whenever you're going to move, the synapse that's made between the efferent output and the muscle cell is cholinergic and involves an ionotropic receptor. These receptors are called actually nicotinic receptors. And in the drugs class, obviously, we go into much more detail about that. But nicotine is, a drug that will bind selectively and activate the inotropic cholinergic receptors. It doesn't do anything to the metabotropic receptors for acetylcholine, but it certainly impacts and activates the ionotropic, the nicotinic receptors. That's why we call them nicotinic receptors. So a nicotinic cholinergic receptor is an ionotropic receptor that binds acetylcholine, opens it turns out like a sodium, and in some cases a sodium and calcium current, but both depolarizing, right? So it'll depolarize the muscle at the neuromuscular junction and generate contraction of the muscle. There are also nicotinic receptors found in the central nervous system. Acetylcholine is really interesting. So there are the nicotinic receptors, right? And then there's also the metabotropic receptors. These are actually called muscarinic receptors because there's another chemical called muscarin that will selectively bind and activate those metabotropic receptors and provoke the release of a G protein upon binding, just like acetylcholine does. So this is actually an interesting aspect of these neurotransmitters, right? That you have the neurotransmitter acetylcholine, right, will act at all the acetylcholine receptors, whether they're ionotropic or metabotropic, whether they're the nicotinic ionotropic ones or the muscarinic metabotropic ones. But you can find chemicals in the environment, right, or that are synthesized that will selectively activate some subpopulation of those receptors. And that's why nicotine, can identify and activate selectively the ionotropic, acetylcholine targets, while muscarin is another chemical that can selectively activate the metabotropic cholinergic receptors, which we call muscarinic. Now, neuromuscular junction in the periphery, very important cholinergic synapses. But in the central nervous system, there is a brainstem source. So there's a nucleus in the brainstem. It has a very long name. It's called the pedunculopontine tegmental nucleus. But we abbreviate it, and you can remember it as the PPT. So the PPT is a nucleus in the brainstem that has a lot of cholinergic cell bodies, right? And their axons will project out. And one of the primary targets for those projections, axonal projections from the PPT here, projecting out, is a structure called the thalamus that we'll see is really important for initially processing sensory information from your eyes and your ears and your body and everything else before sending it on to specific networks and cortex. And this is the projection, this PPT to thalamus connection, sort of becomes selectively active during REM sleep when people are often experiencing dreams. It seems to wake up the thalamocortical kinds of connections or contribute to that, activation. But there's another interesting source of acetylcholine in the cerebrum, and that's actually in, the frontal lobes, sort of deep in the frontal lobes, an area called the basal forebrain. And there's a number of nuclei, deep nuclei in your frontal lobes here, that, one of them is called the nucleus basalis. There's a bunch of them there. But they have, their cell bodies are located in those subcortical nuclei, but they project kind of widely around the brain. But a major target of these cholinergic projections in the central nervous system is the middle of the temporal lobes, which we're going to see are really important for memory function. So they project, from the basal forebrain, from nuclei in the basal forebrain. That's where the cell bodies or soma of these cholinergic neurons are localized. They project, sort of swing around and go into the hippocampus and other structures that are found in the medial temporal lobe. So the acetylcholine is modulatory neurotransmitter. It's the most classical of all the neurotransmitters. It modulates your level of arousal when you're awake and alert. You will have cholinergic firing. It's very, very important to the periphery for the neuromuscular junction, for moving your body around. And in the central nervous system, it plays really important roles in cognition and particularly in memory. It selectively pops back on during REM or sort of dreaming typically associated sleep.